Hello all! I thought I would show you my new office space since this whole working from home. We bought this cool couch because it faces out our window towards the windows and we're so excited about living by the mountains. And when I bought it, the people selling it were pretty excited. It was super cheap. And they're like, look, the seat, it actually lifts up. You can store your guns in there. And I'm from the Midwest. That was a bit of a shocker. I was like, I think I'm just gonna put some magazines in there. And then also we've got a little basket that I put all your assignments in that I grade. My cats have found that that is the perfect sitting spot as well. So what's your space look like? All right, so today we are going to go over chapter 20. Now normally I get 2 days to discuss this chapter because there's a lot of material, but because we missed class due to the coronavirus, I have to try and fit it into one lecture. However, I have dropped some of the material out of it so that there's not quite as much for you to cover. All right. So this chapter is really about genetics of populations. So when we've been talking about natural selection, favoring certain traits over others, we're talking about genes and alleles. So we're going to get into all of that today and a little bit more of how natural selection works and also how you can observe it happening out in the world. Right, so the first objective will be to understand how genetic variation occurs in a population. So how is it that we have, for example, a variety of skin color, or animals can have a variety of heights or number of spots, right? And how, again, why that genetic variation is necessary for evolution to happen. Next, we're going to talk about a principle called Hardy-Weinberg equilibrium and what its implications are for population genetics, how we can use it to predict what frequencies of different traits we're going to see in a population, as well as whether or not evolution is happening. The third objective that you should know is the 5 agents of evolutionary change. So what are the 5 things that can induce evolution? Next, you should understand this idea that a trait that gives a individual greater fitness, that is usually going to be selected for, and it's going to increase in frequency in that population. So we're going to talk about how it is that that works. All right, so why is it that if a trait is more advantageous, you end up seeing it more in future generations? Fifth, you should be able to compare and contrast the 3 ways that natural selection can promote variation. And then sixth, you should be able to compare, define, and compare disruptive, directional, and stabilizing selection. And you might notice that there is a little bit of overlap amongst these different chapters, right? That's okay. They're all really interconnected, right? So let's talk about this first objective, how genetic variation occurs in a population, why it's important in order for evolution to happen. Here we have some examples of what we mean by genetic variation. So within a species, you're going to have some variability in traits, right? So here we've got different beak sizes in birds. We have in the middle different patterns of stripes and colors and sizes in these shells, in these mussels. And then an example in human population, right? We have different features such as hair color, eye color, hair texture, skin color, height, right? All these different things. These are all variations that have a genetic component. So genetic variation is the raw material on which natural selection can act, and the idea is that natural selection is what leads to evolution. And if you remember, we talked about in the last lecture the 3 important conditions for natural selection to occur. Do you remember them? The first one was that variation has to be present in the population, right? So if you're going to say that one trait is better than another, you have to have a variety of traits to choose from, right? For natural selection to occur, it has to have options to select from. So in this chapter, we're really talking about how does that variety, how do those variety ranges, how does that range of selection come to be, right? And when we're talking about genetic variation, a little more detail what that means. So we've got traits, features, behaviors that have a genetic component. And we're going to talk about that a little more of what that genetic component is. You're going to learn what alleles are, right? And how you can have variation in these alleles among individuals in a population. So a little bit of background of kind of some cell biology, right? So parents don't pass on their physical traits directly to their children. Instead, they pass on the directions to make those traits, right? So I pass on the directions for making blue eyes to my imaginary child, right? Those traits, those directions for those traits are called genes, right? DNA. And each person has received one copy of each gene from each parent. So for example, if there's a gene for eye color, you're going to get one copy of that gene from each parent, right? These genes, again, I said they're made up of DNA. They're contained on chromosomes, and you get one chromosome from each parent. So on the left, we have a cartoon that helps to better understand that, right? So focus at the point where we have egg and sperm, right? You'll note that there is one chromosome in the egg and one in the sperm, and upon fertilization, that zygote has two chromosomes, right? One from dad, one from mom. And so now there's directions for creating a person all in there, and there is a set of directions from both parents, right? So not all copies of a gene are identical. So say for eye color, I get one gene, one copy of the gene from dad, one copy from mom, all right? They might not be the same, right? My dad, it's horrible, I don't know what color my parents' eyes are. Let's just say that my dad's eyes are brown and my mom's eyes are blue. I think they both have blue eyes, but anyways, say my dad's eyes are brown and my mom's eyes are blue, right? So they both have the same gene, but they have different variants of that gene. So my dad's passed down the brown-eyed variant of that gene, and mom has passed down the blue-eyed variant of that gene, right? So I have two genes, and there's some variation. All right, so when we talk about these variants, these alternative forms of a gene, those are called alleles. So in this example, let's look at flowers. All right, so here we have a chromosome, two chromosomes, one from each of the parent plants. All right, and on a chromosome, there are tons of genes. All right, one chromosome can have hundreds, tons of genes on it. And so we're just looking at one specific gene on this chromosome, the gene for flower color. Right, and so that's what's shown in that little stripe on the chromosomes. And so one parent has provided the gene that is a variant that produces purple flowers on top, and the other parent has provided a gene with the variant for white flowers on bottom, right? Again, each of those chromosomes, one came from each parent, and now in that offspring, it has both of those chromosomes. It has both alleles, both variants of that gene. Now, if that offspring has two identical alleles, say that both parents had purple flowers, they both passed down genes with the variant that determined for purple flowers, that individual is called a homozygote, right? Homogeneous, right? Same. And if they had two different alleles, then say if they had, in this case that we're seeing here, this would be called a heterozygote. Now, just because you have a certain allele, a certain variant for a gene, doesn't mean it's going to be expressed, right? So in this plant, it has, of the gene for flower color, it has one allele for purple and one allele for white, right? What color is this offspring's flower petals going to be? Is it going to be purple? Is it going to be white? Is it going to be a mixture, all right? So in the case of heterozygous individuals, where the individual has two different alleles for the same gene, like in this situation, only one of them is going to be expressed, right? The other one is going to not be used. So in this case, the offspring is either going to have purple flowers or it's going to have white flowers, right? It's not going to be a mixture. And the way that it is determined which allele is the one that gets used to bring about that feature in that individual is this system of dominant and recessive alleles, right? So one is going to be considered dominant and the other is considered recessive, and the dominant one tends to win out. So in this case, scientists have determined that the dominant allele for the gene for flower color is the purple, right, in this example. And the recessive allele is the allele that codes for white petals. Now, if you have a heterozygote, like we have in this case, if you look in this middle example here, capital A, little a, what's going to happen? The capital A, the dominant allele, which is the purple allele, is going to win out. And so these individuals' flowers are going to result in being purple. Now, if you had a homogeneous individual, say both of the alleles were dominant, in this case, that it would result in purple flowers. And the only way that you're going to get white flowers is if both alleles are recessive. So let's look at this example. All right, we have two heterozygotes that are going to mate. What will the offspring result in? Right? What are the potential color combinations that we can see in the offspring? Now this point here that I want to make, the two alleles remain distinct. They neither blend nor alter each other. That's what I was talking about on the previous slide when I said that you're not going to have, say, light purple or pinkish flowers. It's either going to be purple or it's going to be white. You're not going to see a mixture. Right, so we probably have in this situation some possibilities for purple flowers or some possibility for white flowers, and that's it, right? But how many of them are going to be homogeneous and how many of them are going to be heterozygous, or sorry, homozygous or heterozygous, that's what we're going to figure out next. All right, so we have our familiar Punnett square. All right, so this is going to tell us what the outcome is going to be of this cross. So we have two heterozygous parents, and we're going to use this Punnett square to determine the potential traits of the offspring. So let's start with the top left square. We're going to take one allele from the parent on the left, which is capital A, and one allele from the parent on the top, which is another capital A. Right? So we have a homozygous dominant offspring here. Top right, we take, again, that same allele from the left parent, and this time we're taking the lowercase a, the other allele, from the top parent. This is heterozygous. Let's do the bottom left. All right, we're going to take that lowercase a from the parent on the left and the capital A from the parent on the top. Again, we have heterozygous offspring here. And then for our last square, we're going to take the lowercase a from the parent on the left and the lowercase a from the parent on the top. And so that is homozygous recessive. So the results that we have, if the offspring is two capital A's, we said that is homozygous dominant, and that offspring is going to have purple flowers. Then we have two options here where we have heterozygous offspring, and it doesn't really matter what order the A's are written in. But we've determined that the dominant allele is the purple allele. So if the offspring are heterozygous in this case, they are going to have purple flowers. And then our last square are homozygous recessive offspring. They have both of the recessive alleles, and so in this case they are going to have white flowers. Right? So this is the percentage or the frequency that you're going to see of the different offspring. So we're going to have 25%, or 1 out of 4, of the offspring are going to be homozygous dominant. 2 out of 4, or 50%, are going to be heterozygous. And 1 out of 4, or 25%, are going to be homozygous recessive. And you can see that there's a typo here on the top left. This next to white, this should say lowercase a, lowercase a. So I apologize for that. But go ahead and try to do this on your own. Make a Punnett square, fill it out, write out how many you get of each type, figure out how did I get 25%, how did I get 50%, all right? Now I've been using the word phenotype in this and previous lectures, and hopefully you've looked that up, but I just wanted to make sure that we're all on the same page. Phenotype is just a way to say the physical appearance of an individual, some specific trait that is observable and that has a genetic component, right? So the ability to roll my tongue is, I think people said that that has a genetic component, right? That's the phenotype. The gene that allows me to do that is the genotype, right? Something like the ability to do math, all right? That's a learned trait, right? That's not a phenotype because there's not a gene that's responsible for my ability to know that 2 + 2 is 4, right? And so again, these different examples of phenotypes that we saw at the beginning, right? There's going to be variability in these phenotypes. So for eye color, that is a phenotype. There's going to be variation in the human population, brown, hazel, blue, green, right? Again, genetic variation is the raw material that natural selection can use in order to pick a certain trait and to have evolution occur. Let's look at an example in giraffes. On the right, we have a population of giraffes, and the trait, the phenotype that we're looking at is height, right? So height has a genetic component to it, and there's some variability in this population. Some of them are a little bit taller or have, I guess, longer necks than others, all right? And this is just natural, right? There's some variability in this. Now, again, this is has a genetic component. So parents that have really long necks are going to pass on long neck traits to their children. Those with short necks can pass that trait on to their offspring as well. Now, we've got variability, right? Condition number 1 for natural selection, and we also know that this variability is hereditary. So that was our third condition for natural selection. Our second condition was what? It was that variability has some of those traits are beneficial or harmful to the survival of that individual. So in this case, having a longer neck is beneficial. It's providing more opportunities to get food, more resources, now more energy. That animal is more likely to survive than a shorter animal. And so natural selection is going to favor the individuals with the longer necks. Life is hard out in the wild, right? If an animal is too short, if it can't get enough food, it's not going to be as big, it's not going to probably get as many mating opportunities, it could even starve. However, the giraffe that has the longer neck, right, it can eat more, it's going to be stronger, it's going to be more attractive to mates. If it's a female, it can invest more energy into caring for its young and provide better for its young. And so it's more likely that its genes for long neck are going to get passed down than the traits for short neck in this example. So what we're talking about in this chapter is called population genetics. That is looking at the mechanism of how traits can be passed on, how they can evolve over time in a population, specifically due to the variability in genes, right? So we're looking at what percentage of these different alleles are present in a population, and how these genes, how these percentage of alleles are changing over time. So for example, in population genetics research, you could be looking at, in that case of the peppered moths that we talked about earlier. So scientists found that different environments favored lighter colored versus darker colored moths. And we knew that that color property had a genetic basis. There was a gene responsible for moth color. And so there are alleles for that gene that promote lighter colored moths and alleles for that gene that promote darker colored moths. So in population genetics, you're tracking the color of the moths and also the percentage of homozygous dominant and recessive and heterozygous individuals, and looking at how do those frequencies change over time. So a little bit more about population genetics. An important concept or important rule in population genetics is that of the Hardy-Weinberg equilibrium, right? And so we're going to talk first about what Hardy-Weinberg equilibrium is, and then what it can be used for, right? So for example, in our case of the flowers with the purple or the white flowers, we said that the purple flower allele was the dominant allele. And so you may wonder, how come over time, over many generations, the white color doesn't just go away, right? Why is it that there's still plants out there with white flowers? So the Hardy-Weinberg principle explains this. And it determines that the original population, the proportions that are present at the beginning of a certain time frame, even after many generations, they will stay the same. All right, so as long as the environment doesn't change, as if evolution is not occurring, you can predict how many white or purple flowers you're going to see in future generations. Now, I don't expect you to know the equations related to the Hardy-Weinberg principle or how to complete the equations. I would rather that you understand what is the principle and what can it be used for. All right, so first, it's explaining why or how. It's a method of determining the proportions of genotypes in a population over time. All right, you can predict what percentage of individuals in a population are going to have certain traits versus others. Again, this is in a large population. If evolution is not happening, if mating is random, right, you can use this equation to predict frequencies in future generations. So for example, let's talk about fur color in cats, right? Say there's a gene for fur color determining whether the cats are going to be black or white. You can go out, find a population of cats, count the number of black and white cats, and use this equation, this principle, to say the next generation of cats is going to have this many black cats and this many white cats, right? So let's look at this equation or this principle in use. All right. So in the case of cat fur color, we're going to say that the black allele for fur color gene is the dominant allele, and the recessive allele for fur color is white. Right. So we have one gene for fur color, and we have two variants. One is dominant, that's going to result in black fur, and the other allele is the recessive allele, that's going to result in white fur. And you go out and you count, say, 100 cats, and you find that 16% of the cats, or 16 of the cats, are white, and 84%, or 84 of those 100 cats, are black. Great, all right? But which of those black cats are homozygous dominant and which are heterozygous? That's important to know, right? If you're going to make predictions about future generations and determine if any natural selection is occurring. And so you can use this equation to determine that, and using the Hardy-Weinberg equation, you can determine that 16% of, we said that 16% are homozygous recessive. We can determine that 48%, all right, are homozygous black, and 36% are homozygous black. Again, using the Hardy-Weinberg equilibrium, when we've got a population of white and black cats, we know that 16% of them are white and the remaining are black, but we don't know which of these black cats are heterozygous or homozygous. So using this equation, you can determine that, say, out of 100 cats, that 36 of them are homozygous dominant for the black color, and that 48 of them are heterozygous and have the black color. All right. Again, you can use the Hardy-Weinberg equilibrium to predict genotype frequencies. You can say, in the next generation of cats, we are going to see this many black cats and this many white cats. But this is only true if evolution is not occurring. Right? So say you follow that population of cats and you go to the next generation and you count up all the cats, and you find that the actual number of cats that you've seen of the different colors isn't matching what you predicted. All right, this is really important to do if you're studying natural selection. Right, you have this prediction. You say, if natural selection is not happening, if evolution is not occurring, these are the numbers that we should see. So if you go out and you find that the numbers are not what you would predict, that is implying that evolution may be occurring in that population, right? So the Hardy-Weinberg equilibrium can be used first to predict what would be future genotype frequencies in a population, and also to determine whether or not natural selection could be at play in that population, all right? So again, we can use this to determine the normal amount of genetic variability in a population. And if the population is outside of that normal range, you can start to look at the possibility of evolution occurring. So say in our example of purple and white flowers, say you look in future generations and you say, all right, we should expect to see, you know, this many purple and this many white flowers, and you're starting to see that there's more white flowers than you expected. You say, okay, something's happening. Maybe there's something about white color that is allowing, that's giving an advantage to those flowers versus the flowers with the purple color, right? Natural selection, evolution at work. So if Hardy-Weinberg equilibrium is not present, if your population is not matching the predicted genotype frequencies that you calculate, that suggests that evolution could be occurring. And so in order for equilibrium to be present, you have to have these following rules, all right? So for a population to stay in equilibrium, there should be no mutations happening, no gene transfer, all right? So no new individuals coming into that population, bringing new varieties, or no individuals leaving the population. Third, mating must be random. Fourth, the population must be sufficiently large. And fifth, natural selection should not be happening, right? So for a population to be in equilibrium, none of these things can be happening: mutation, gene transfer, selective mating, small population, or selection. So you go out and you find that your population is within Hardy-Weinberg equilibrium. You can assume that none of these things, that all of these rules are being kept. However, if you go out and study a population and you find that it's not in equilibrium, you can say maybe one of these 5 things is happening. Maybe a mutation occurred. Maybe there was selection for some trait. Maybe one of these traits is providing an advantage over another. So those 5 rules, or those 5 conditions, those are what make up the 5 agents of evolutionary change, right? So if you're saying that we think that this population is undergoing evolution. It could be happening in these 5 different mechanisms. Again, the first one we talked about was mutation. The second was gene flow. All right, so this would be where you've got kind of like interbreeding between different populations. Next, non-random mating. So there is some trait that maybe is preferable, and that's any individuals with that trait are getting to mate more often than others. That's non-random. Another example is self-fertilization, and this happens in plants, right, where the pollen from a plant fertilizes its own stigma. Next, we can have genetic drift. So if a population is too small, any slight anything that happens to that population to change the percentage of different traits is going to have a really drastic effect. And then lastly, we have selection, right? So a process in which a certain trait is giving an advantage over other individuals without that trait. So I'm going to go into these 5 agents in more detail, right? The first is mutation. Now mutation, again, this is where the DNA sequence gets changed, right? And this is the ultimate source of genetic variation. So when we talked about having a gene for flower color that has 2 alleles, purple and white, how do those alleles even come to be, right? Those alleles formed because at some point there was a mutation. And so now you have 2 genes that are very similar in sequence, but there are some changes between them due to mutation. And that is what results in one gene coding for purple flowers and another coding for white flowers, right? So it makes evolution possible. However, mutation rates are generally pretty low, right? It's not like you're going to see, oh, look, we've got a pink flower, every year. Or there's not going to be like a ton of mutation happening. But it does happen, and that is what causes that variability in a population, right? Next, we have gene flow as an agent of evolutionary change. Say you have a population, 2 different populations, and they're pretty different, and an individual from one population travels to the other and mates with an individual in that other population. Now it's bringing in some new alleles, some variety to that population, right? In the example in this picture, we have a bee traveling from a population of purple flowers to a population of pink flowers. So in that purple population of flowers, it probably didn't have very many alleles for pink, if any, for pink color, all right? And same in that pink flower population. They probably had very few to no alleles for purple. But if a bee travels to a purple flower and gets some of the pollen, right, that is a gamete that contains DNA on its body, and then it travels to another flower patch of just pink flowers, and some of that pollen falls off onto that pink flower. Now, when that fertilization occurs, we now have introduction of some new alleles, these purple alleles. And now you might see in future generations in that pink population, there's some purple flowers mixed in, right? Next, we have non-random mating. So non-random mating just means that there is mating occurring, and it's happening kind of because maybe there's a trait that is more beneficial or more attractive. So you could have, for example, on the right, we talked about how in birds, these tropical birds, that there's certain traits that are attractive to the females, such as bright color or long tails, right? So that means that some of the males are going to get to mate more frequently than other males. That is non-random mating, right? Also, you can have things, like I said, self-fertilization. That is not random, right? And You can also have things like where an individual is more likely to mate with another individual that looks similar. All right. So when that happens, that is called assortative mating. Right. So individuals, a male and a female that look more alike, maybe for some reason that is attractive, and so they mate more frequently. What this is going to do to a population is it's going to increase the proportion of homozygous individuals. Right? We're kind of reducing the variety because only like individuals are mating with each other. The opposite is called disassortative mating, where phenotypically different individuals mate. Right? So they don't necessarily find an individual that looks similar to them attractive. They want something that looks different. And in that case, then you're going to promote more heterozygous individuals in that population. Right? So non-random mating. The fourth agent of evolutionary change is genetic drift. And hopefully you watched the video that I posted in a previous lecture about genetic drift, but this chapter gets into it in more detail as well. So in genetic drift, this is only happening in very small populations, right? In a small population, allele frequency may change by chance alone. What do I mean by that? So let's look at this example in our picture on the left. We have a population of birds on the mainland, and they have variability in feather color. They could be red or yellow, right? And a subpopulation travels to an island and stays there and mates with each other, right? So if a very small subset of the mainland birds left, in this case, for example, only 3 left, and it just so happened that none of the yellow birds left, right? And so there are no yellow birds on that new island, and over time, red is going to predominate as the feather color in these birds. So often this can also be something such as a catastrophe or like an act of God can happen, right? So if you have a very small population and something happens, right? Maybe you have a small population of trees and there's not a lot of variability in that and a forest fire happens, right? There's a greater chance that some of those alleles are going to be lost forever in that small population. We're going to go over some examples of this as well, but you should make sure that you understand this concept. So the influence of genetic drift is dependent upon population size. So the smaller a population is, the more at risk they are for genetic drift. Right? So the key thing to remember about genetic drift is that it's more likely to happen in small populations. Genetic drift can be split into 2 types called the founder effect and the bottleneck effect. So here again we have in that picture what I demonstrated was an example of the founder effect, right? So only a few individuals disperse and become the founders of a new isolated population, all right? Let's talk about like humans, all right? Say you have back when the Puritans came to North America, all right, only a small percentage of the Puritan or English population came to North America. And so what if, say, nobody came over that had brown eyes? I mean, that didn't happen, but say if in that small population on those ships, none of those people had brown eyes, or maybe none of them could wiggle their ears, all right? Generations later, those traits are going to be lost forever, right? Again, on this example, a lot of times we're talking about kind of like pioneers, founders of a new population in a new environment. A great example is on islands. All right, so you can also watch, there's plenty of videos online about the founder effect and about genetic drift that you can watch to better understand this concept. The other type of genetic drift is called the bottleneck effect. So when I talked about natural forces or catastrophes, right, that's tending to relate to this bottleneck effect. So in this case, you have a small population not because they left and went to found a new population somewhere else, but that something horrible happened and the majority of the population is gone, right? So whoever is left behind is whatever genes they have are what future generations are going to have as well, right? So this is going to, again, result in a loss in genetic variability. Let's look at an example. Here we have in seals, right? So a long time ago, there used to be a variety of different fur color in seals, as we can see on the bottom left. In the late 1800s, people began hunting seals very strongly, and they really greatly reduced the population of seals. And so only a small fraction of seals were remaining, and only the ones that survived are able to pass on their traits for fur color. So it just so happened in this example that the tan and white seals are the only remaining seals that were left to reproduce. And so that surviving population, when they reproduced, future generations only were tan and white because that's all that was left to reproduce, right? An important thing about the founder and the bottleneck effect is that it is random genetic changes, right? So it's not like the white and the tan seals had better survival, like it's not like they were faster or better able to avoid humans. It's just random. Like they just happened to be the ones that were left over after this extensive hunting by humans. So why do we say bottleneck, right? Here is kind of a more abstract representation, right? So when you have a large population, this parent population, you've got variability. And in this example, the most common color is blue and yellow, but there is some red mixed in, right? So when there is a bottleneck, a drastic reduction in population, right? So this bottleneck in this bottle, when you try to pour out some of these beads, only a few come out, right? So now we have a very, very small percentage of individuals surviving. It's not very likely that those surviving individuals are going to have that red color, right? Because red was pretty infrequent to begin with. And so only who is left over, only the individuals that are left over are the ones that get to reproduce. Only their genes, their traits are the ones that are going to get passed down. And so after that bottleneck, after that drastic catastrophe or whatever it is that happened, in future generations now we have completely lost that trait for red color. So let's talk about the fifth agent of evolutionary change, which is selection, right? So this is one that we've talked about before, right? It's a pretty commonly discussed method of evolution. And in this with selection, you have a trait that is conferring some type of advantage over individuals versus others. So this can be artificial selection, such as breeders selecting for certain characteristics, such as maybe selecting for cows that can make a lot of milk, right? Or dogs that have a very docile behavior. And it can also include natural selection, where the environmental conditions are determining what individuals produce the most offspring. So in this cartoon example, we have a genetic trait, so gene relating to caterpillar color. And in this environment, the yellow color is disadvantaged, is not advantageous because the yellow caterpillars cannot camouflage and they are more likely to be preyed upon. So future generations of caterpillars are more likely to be green than yellow. So this is an example, that caterpillar example is selection to avoid predators. And here we have another example of natural selection to avoid predators seen in pocket mice. So they're called pocket mice because they like to shove food into their cheeks and they pouch out kind of like pockets. They're pretty cute. And in the next lab, when you submit your final paper, we're going to take a little break and you guys are going to get to watch some fun videos about natural selection, and you're going to watch a video about these pocket mice. So in this case, the variability, the trait or the phenotype is fur color. Right? This fur color has a genetic component, and there was variability in fur color in pocket mice. Some of them were black, some of them were lighter. And in this case, having a lighter color was not good. So when these pocket mice live in locations where there's a lot of volcanic rock on the ground, that volcanic rock is black, and so the lighter colored mice stick out and they are more easily seen by predators. So if you get eaten, can you mate and pass on your genes? No. And so in that environment, the light color is not going to be selected for, it's going to be selected against, and future generations are going to have more and more alleles for that dark fur color. Now on the opposite side, in other parts of the desert where there's not this volcanic rock, there's light sand, and so having a dark fur color is actually not beneficial, right? And so in future generations in that location, you're going to see more and more individuals with the light colored fur. So you can also have, in addition to selection that's helping to avoid being preyed upon, you can also have selection to help live in a certain climate, all right? So again, we're talking about the second rule of natural selection, in which the environment is playing a role on which traits are beneficial and which ones are not. So one example we can look at is in the species of fish, where they are found at a variety of latitudes. So some of them are found up north where it's colder, some of them are found further south where it's warmer. And interestingly, there's a gene for an enzyme that they share that has two different alleles. So one allele for this enzyme is going to function better at a cold temperature, and the other allele for this gene is going to code for an enzyme that functions better at a warmer temperature. Right? So you have one gene for an enzyme, two variants, two alleles, colder temperature optimized and warmer temperature optimized. And this enzyme happens to be important for metabolism for the fish, so kind of taking care of how they digest their food and get energy from their food. And so what you find is that when you look in these species of fish up in the north, you're more likely to find that allele for the gene that is north optimized that works better at colder temperatures, right? Because it's colder up in the north. And when you look at the function of these fish, they actually can swim better with this variant. So if you have any fish up north that have the other variant, the warmer temperature optimized enzyme, they don't swim as fast. And so it's likely that this allele is providing them with some type of advantage, selection, and selective advantage. Next we can have selection for pesticide and microbial resistance. This is kind of like selection for avoiding predators. So in this example, we have flies, bugs, that are over time evolving to avoid being killed by pesticides, which is no fun for us, right? So human scientists have come up with a variety of different insecticides, pesticides to help kill bugs, whether it's out in your fields or in your house, in your neighborhood, right? And so over time, certain individuals of these bug species that have some type of allele that provides a resistance are going to increase in number. So we have actually seen the evolution of insecticide resistance in over 500 species of bugs. And we're going to look at one example in housefly that is able to resist insecticide. So there are two genes that can provide resistance. One is called PEN, and one is called KDR, and actually there's a third, DLDR. I don't expect you to memorize these genes. I just want you to be able to understand that there are genes that can allow for pesticide resistance, and we'll also talk about microbial resistance. So in these flies, when they're exposed to a pesticide, that pesticide has to bind to a transporter on the surface of the cell in the body of those flies. And if that pesticide can enter into the cell, that fly is going to kill the cell, that fly is going to die. So all of the flies have the gene for this transporter. However, some of them have a variant of this gene that for whatever reason won't transport that pesticide. So you've got an original population with some variety. Some of them will take up the pesticide, some of them won't. And when a lot of them get exposed to this pesticide, all of the ones that will transport it, what happens to them? They die. Do they get to pass on their genes? No. But the subpopulation that has a variant of this gene for the transporter that doesn't take up pesticide, they survive. They get to pass on their genes, and so over time, more and more of the individuals in that population will be resistant against that pesticide. So that is an example of PEN, right? The PEN gene. Another example in which bugs can resist pesticide is that they could just have fewer of these transporters. Right? So maybe so you're having, in this original population, you have variability in how much of this transporter is present in those cells. Right? So some of the individuals have a lot of transporters, others have fewer. Right? So if you have more of these transporters in your cells as a fly, are you going to be more likely to survive or to die when you get exposed to pesticide? Those flies would be more likely to die, right? And to not be able to pass on their genes. However, other individuals in that population that have fewer of these transporters, less of that pesticide is going to get into their cells and they are going to better survive. They're going to get to reproduce and pass on their genes, and their offspring will also have fewer of these transporters. So these are just two examples, two ways in which pesticide resistance has occurred. Again, you've got some type of change in the environment, in this case, exposure to pesticide, and that is changing and that's determining what traits are beneficial and which ones aren't. And over time, that's affecting that population. Another example that we have is evolution of resistance to antibiotics, right? This is really important for human health. So we have bacteria that attack people, right? They attack our cells, and scientists have come up with these antibiotics, these drugs that will kill bacteria. However, just like in us, in bacteria, there's variability in their traits. And so within that population, there might be a small percent that have traits that allow them to resist that antibiotic. What happens? Every bacterial cell that doesn't have that trait dies, but the ones that do have that resistance gene, they survive, they pass on their genes, and over many generations, you're going to see more and more of the individuals in that population have that resistance gene. So that is why your doctors nowadays are trying to prescribe less antibiotics. They don't want to encourage the bacteria to evolve and to get a resistance against the drugs. So they're going to use it less frequently, but when they use it, it's going to be much more effective. So let's talk about why a phenotype that brings greater frequency is going to end up being seen more commonly in future generations. Right? It's our fourth objective. And we've talked about this before, right? This idea of fitness. We're just going to go into more detail of this concept today. So fitness, we said, it's not necessarily having to do with how fast you can run a mile, right? Or how many chin-ups you can do. I can do 0. But it has to do with your ability to leave behind more offspring to the next generation, right? And why is this important? Because these traits are passed down to our offspring by genes, right? So if you can pass down your traits, that's great. If a trait results in better fitness, that gene is more likely to get passed down, right? So fitness, again, the ability to leave behind more offspring that are more likely to survive. So fitness can encompass things like traits that just allow an individual to survive, right? If you're dead, you can't reproduce. It could involve traits that allow an individual to be more successful at mating. Again, the more chances to mate, the more offspring. It could also be related to the number of offspring per mating. So if an individual is healthier, it's eating well, it's got lots of resources, it's probably going to have more offspring every time it mates. And traits favored for one component may be a disadvantage for others. We've talked about that in the case of like birds and sexual selection, how they, in the tropics, right, they're evolving, they're adapting to have these big giant tails, right? That's great for sexual selection, but maybe not good for survival. So there's kind of a bit of a balance, a back and forth amongst these different traits. Again, whatever individual has the greatest fitness, those individuals are going to get to pass on their genes, and their genes are going to be seen in a greater proportion in future generations. So here we have some examples of fitness, right? On the right, we talked about sexual selection. So in this case, this bird is very fit because it is very attractive to the female. In the middle, we have an example of fitness in terms of survival. So in bug color, certain colors may be promoting more camouflage ability, right? And so the individuals with colors that don't allow them to camouflage get preyed upon. They don't get to pass down their genes from that color. And so in this case, over time, you're going to see more brown bugs, right? Another example is the number of offspring, all right? So if you would think, oh, the more eggs this bird lays, the better fitness, right? It's passing on its genes to more offspring. That's awesome. But we talked about how some of these different, these traits, sometimes they conflict. So if a robin lays a ton of eggs, those offspring are probably not going to be as well taken care of as a robin that lays fewer eggs. And so there's going to be a balance, a selection for a certain number of eggs. For that species, there's going to be an optimal number of eggs that it lays, that it can take care of well, and that these offspring are likely to survive. And so we can see that over time, the range in the number of eggs that was originally seen in blue is going to narrow down because it's getting optimized. Let's talk about promoting variability, right? So natural selection, normally we think of it as reducing variability, choosing a certain trait that's beneficial, but natural selection can also promote variability in a population. So again, normally we think of natural selection as removing variation because it's favoring one trait over others. For example, in horses, we talked about in the evolution of horse species that originally the horses lived in very forested environments, and then over time, due to climate change, these environments switched to grasslands. And so in a grassland environment, it was better for these horses to be able to run really fast, right? A predator can see it from far off. It's got lots of room to run. And so over time, these horses are going to adapt to have longer legs, right? And over time, the majority of the alleles in that population will be for long legs. So that is reducing variation. However, in some situations, selection can do the opposite and can promote variability in a population. So we're going to talk about some examples in which that can happen. So the first way that variability can be promoted in a population is a method called frequency-dependent selection. Right? So this is selection dependent upon the frequency of a trait in a population. You can have negative and positive frequency-dependent selection. So in negative frequency-dependent selection, rare phenotypes are advantageous, right? So in this case, you're going to be promoting these rare phenotypes, these rare alleles. You're going to be promoting more variability in the population. So for example, maybe a predator is searching for some bugs and it has a certain picture in its head of what this bug looks like that it wants to eat, right? Like, I'm looking for the tan bugs with 5 spots. Those are my treat for the day, all right? So if it sees a rare form of that bug, maybe it doesn't have spots, it's probably not going to eat that bug, right? It's not what it thinks of when it's searching for prey. And so it's going to survive. It's going to survive better than its counterparts. And that trait is going to get passed on into future generations. And you're going to still have some variability in that population. The other example we talked about, positive frequency dependent selection. In this case, whatever is most common is the trait that is most advantageous. Right? So in this case, the oddballs stand out and they're more likely to get eliminated. So let's look at some examples of these. Here we have an example of negative frequency dependent selection. Right? So in this case, we said having a rare phenotype is beneficial. Right? So we have these bugs called water boatmen, and they come in a variety of colors. So there's variability in the population in terms of color. And when you have an equal amount of the different colors, say we are looking at dark brown, medium brown, and light brown. You have, a third of the bugs are dark, a third are medium, and a third of light. Whatever color provides the best camouflage is the one that's going to be selected for. But if you have a population where one of these colors is very popular, such as, say, dark brown, that color is going to be, that colored bug is more likely to get eaten by prey, right? So fish will eat the most common color type more than they would by chance alone. So what's going to happen? The lighter colored bugs are going to survive better, and their genes are going to get passed down into the offspring, and we're going to be promoting variability of color. So again, originally you have a situation, if you have equal numbers of colors, maybe brown is a good color and it is beneficial because it's a good color for camouflaging in a brown environment. All right, so maybe selection is going to choose that brown color, and you're going to see reduction in variability in future generations, and eventually you're only going to have dark brown boatmen. However, if you have a population where dark brown is the most common color, now those boatmen are going to be eaten more frequently by fish. So now what's going to happen is that those alleles for lighter colors are going to stay, they're going to increase in that population, and now we're starting to increase variability in color in this boatman species. And so that's what this graph is showing. On the y-axis, we have percent of color type taken by fish predators. And on the x-axis, we have color type frequency and population. So if we have, for example, a high percentage of, let's look at this example, light brown bugs, they are going to be very likely, right, highly likely to be taken by predators. And the same thing with brown, right? If brown is the most common color, then it is going to be most commonly eaten. And the same with medium brown, right? So in this case, it doesn't matter what color you are. Color is not providing any type of camouflage advantage. It is just whatever color is most common is the one that gets eaten and selected against. In positive frequency dependent selection, we talked about the idea that the most common trait is the one that gets passed down. Oddballs stand out, and not in a good way. So in this example, we have catfish eating some fish. These look a lot like zebrafish. And there is one oddball. It is easier to see, it is more noticeable, and so that catfish is like, oh, of course, that's the first one it sees because it's obvious, it's sticking out. That fish is more likely to get eaten than the commonly colored fish. So that is positive frequency dependent selection. So again, just overall taking a step back, frequency dependent selection is a type of selection that is dependent upon how common a trait is in a population, all right? And this is a type of selection that can promote variability in a population. Let's look at another method of promoting variability in a population. This is called oscillating selection, and we've actually seen an example of this before. So in this case, you have over time different traits being beneficial, right? So in our example of ground finches in the Galapagos Islands, there were periods of time where it was dry and other times where it was wet, right? And so in that population, over time, sometimes it was beneficial to have big bills, and sometimes it was beneficial to have small bills. And so what that means is that over decades, you're not going to be seeing one size bill becoming more popular, more common, right? Because what's happening is that you have this oscillation in the environment. So that when the environment is changing a lot, that's going to promote variability. Now, say you had a climate change occur, and for decades you have dry environments, right? That's not oscillating selection anymore, right? Because the environment has permanently changed, and now there is one trait that is definitely more advantageous, having the big bills. And again, if you remember, why is it that big bills were beneficial? During drought, there's not much food around, and if you have a bigger bill, you are more able to eat big seeds. And if you have a smaller bill, you just can't eat those, and so you die because you starve. Sad, right? So if you have the environment changing or selection favoring one phenotype versus another, and it's bouncing back and forth over time, you are going to have a lot of variability in your population, because one trait isn't always the best. Before we talk about our third method of promoting variability in populations, I want to discuss the science mystery, and you're going to watch a video about this as well in lab. So sickle cell anemia is a disease in humans, and this affects hemoglobin, which is an important molecule for transporting oxygen in the blood. And so what happens is people that have sickle cell anemia are going to have severe anemia, so problems with their blood and with their cells are basically not getting enough energy, right? And so people that have sickle cell, the sickle cell allele, usually don't live very long. So we have a gene that is responsible for hemoglobin, and you have two different variants, all right? One is the normal and one is the sickle cell allele. And so if a person is homozygous for that variant, for that sickle cell allele, if they have both of those, they are going to have anemia, not going to be very healthy, and are not going to live long enough to reproduce. Now, currently nowadays, we do have treatments that can help with this, right? But in the past, in the history of human evolution, that wasn't necessarily the case, right? So, interestingly, nowadays we still see people with sickle cell anemia, right? And so you would think, why in the history of human evolution did sickle cell anemia stay around? Why was this genetic variation maintained? So let's look at kind of the history of this disease. So the sickle cell allele is commonly found in people with African descent, right? And another condition, another disease that is common in people that lived in Africa in the past is malaria. Interestingly, historians found that people that had sickle cell anemia that were heterozygous, they were less susceptible to malaria. So people that were homozygous for sickle cell anemia, really sick, not able to reproduce. People that were heterozygous, they had one normal allele for hemoglobin and one was the sickle cell allele. They don't have anemia, but they are also less susceptible to malaria. When we look at this map of Africa, in the regions with the lines, these are regions where malaria was common, all right? And in the colors, shades of red, this is indicating the degree of sickle cell allele presence. What you can find is that in areas where there is a malaria, you actually see more people with this sickle cell allele, right? Areas that don't have malaria, you don't see people with that sickle cell allele. So there's something, some type of interaction occurring here. Now, for sickle cell anemia, the sickle cell allele is a recessive allele, as you may have discerned. So only people that are homozygous recessive, that have two copies of this sickle cell allele, this recessive allele, are going to suffer from the disease and are going to have anemia. People that are heterozygous in that case then are not going to have sickle cell anemia, right? Because in a heterozygous situation, the dominant gene, which is the healthy variant, is the one that's going to win out. But they still have that allele present, all right? Now, for some reason, these people, again, are more resistant to malaria, right? Now, people that are homozygous dominant, that don't have any alleles for sickle cell, they do not have sickle cell anemia, right? And they are vulnerable to malaria. So even though we have a situation where they're heterozygous, the dominant allele is winning out, somehow that recessive allele is doing something. It has some type of slight activity. And so something about that recessive allele is beneficial for survival against malaria. And these people, when they get infected with malaria, they will have sickle cells temporarily, just during that time of that disease. And something about having sickle cells is providing resistance against malaria. So what's happening here, this is called an incomplete recessive allele. In other words, the typical rules that we follow of dominant alleles being the ones that are expressed and recessive alleles being silenced, that's not happening, right? So it's incompletely recessive. Somehow this recessive allele, the gene that it codes for, is being slightly expressed. And in this case, it's being expressed when that person gets malaria. Again, you're going to watch a video about this that will explain this in detail. So what this is called, or what this is conferring, is a heterozygote advantage. So this is the third method that allows the promotion of variability in populations. In this case of sickle cell anemia, being a heterozygote provided an advantage against malaria. And what that means is that gene for sickle cell, that allele for sickle cell, is going to get passed down because those people are more likely to survive in the past. We didn't have medications for malaria or for sickle cell disease. And so that is why nowadays we still see people with sickle cell anemia. There's variability in the population. So for whatever reason, in some situations, heterozygotes can provide an advantage. Usually this is due to this phenomena called an incomplete recessive allele. All right, so this promotes both alleles, the dominant and the recessive, to stay in that population over time. So the third method that variability can be promoted in population is heterozygote advantage, and it's due to this phenomena called incomplete recessive alleles. Our last objective is to talk about different types of selection, different types of natural selection. And you've actually learned about a lot of these already, so this shouldn't be completely new. Now, sometimes you have a trait that is determined by more than one gene, right? So, for example, body height in humans, there's actually a set of genes that determine a person's height, not just one. And so when you have more than one gene determining a phenotype, you're going to have disruptive, directional, or stabilizing selection occur. Selection is going to occur on all of these traits, all of these genes. Here I have a graph that demonstrates these different types of selective processes. So let's start on the right, stabilizing selection, because that's one that we've talked about before and that you're probably familiar with. So we're looking at an example of body size, and body size is determined by multiple genes. And over time, a certain size is going to be selected for, is going to be optimal. You don't want to be too big because then you need to eat a lot. You don't want to be too small because then you aren't strong enough to get resources or to be attractive to mates. And so there's going to be an optimal body size that over time is going to be selected for, in this case, about 75 grams. So that's stabilizing. You're stabilizing, narrowing in on one body size that is best. Now, you may have another situation, be in another environment, where having a larger size is better. So maybe it's worth it to be larger in order to have better chances at mating. Say, you're in a situation where males need to compete with each other, right? Or maybe having a larger body size allows you to crack open larger muscles and get to more food, more bang for your buck, right? So in that case, having a larger size is more beneficial, and so that average body size over time is going to shift, in this case to larger. So if the average is changing over time, becoming greater or smaller, this is directional selection. Lastly, we have disruptive selection, and we've talked about this before as well, right? So in this case, body size, it's not really great to be average. It's better to be really small or to be really large. And I'm trying to think of an example in which that would be good. and I really can't think of it, but say you have this extreme environment where being really small allows you to hide easier from predators, or if you're really large, you can just beat up your predators, right? So being average in this environment is not helpful. You either want to be really big to fight predators and survive, or you want to be really small so you can hide in nooks and crannies and not get eaten. So in this case, what's going to happen is that the most common size is there's going to be two options. So you're going to see that about 50% of the population is going to be really big, and 50% of the population is going to be really small. But a very small percent of the population is going to be average. And so this is called disruptive because now you have two different traits that are beneficial. Let's look at another example of disruptive selection, but not in body size, but in beak size. And I think I've given this example before. So in the environment of the African black-bellied seed cracker finch, there are, the food that they eat is seeds, and in that environment, seeds come in just two sizes, big or small, right? So if you want to eat the big seeds, you got to have a big bill. And if you want to eat the small seeds, you have to have a small bill. And in this case, birds with medium-sized bills, they're not really great at opening either seed type, eating either seed type, and so they actually have a disadvantage, right? So they're unable to open large seeds because their beaks are too big, or not big enough, sorry, and they're too clumsy to process small seeds because their beaks are just too large. So I was told that if I put black oil sunflower seeds in my bird feeder, that they're actually too small for magpies to eat. So that is possible that your beak can be too big to eat a small seed. So in this case, in this environment, it's good to either have a really big beak or a really small beak. And so what are we seeing here? Disruptive selection over time. Next, we have directional selection, all right? And in this example, you're eliminating one extreme, and this can occur when the environment changes. So there's an experiment in which scientists have demonstrated directional selection. So we have this variability in behavior that has a genetic component, and this behavior is tendency to fly towards light, all right? So on the y-axis, the higher the number, the more likely the behavior is that fly has a tendency to fly towards light. If it has a lower number, that fly doesn't have much of a tendency to fly towards light, right? Now, in this experiment, what they did was flies that got too close to the light, they died. Maybe they used like a bug zapper lamp. I'm not sure exactly how they did this experiment, but what happens over time is they tracked the generations and they tracked their tendency to fly towards light. And what we found was that the tendency, this behavior changed, right? So over time, more of these flies didn't care about light. More of these flies tended to not be attracted to the light. Now, this is not a situation where they learned not to fly towards the light. It's not like they said, hey, look, Cousin Vinny, look what happened to him because he flew towards that lamp, he died, we better not. All right? Natural selection has nothing to do with learning or with desiring to have a trait or not. Natural selection, it's tough, it's mean, all right? The bugs that had this behavior to fly towards the light, this behavior had a genetic component, and they didn't get to pass that gene down because they got zapped. And the flies that had the genes that made them not care about the light, they were the only ones that survived. They're the only ones that got to pass down their genes to the next generation. And so that behavior is what you're going to see more likely in future generations. And this is why I always squish bugs in my house, because I'm hoping to promote selection for bugs that learn to not come inside of my house. I don't know if it's working, but I'm trying to do some artificial selection in my neighborhood. Let's look at another example of stabilizing selection. So in this case, being at the extreme is not beneficial, and being average is good, right? So in this example, we're going to look at human birth size, birth weight, right? Now we have an average birth weight of about 7 pounds, right? And there's a reason that is the case. In the past, before we had doctors and medicine and things like that, if the baby was too small or too large, they were more likely to die. And so that gene that determines birth weight, there were different alleles. Some alleles promoted smaller birth weight, lower birth weight. Some alleles for this gene promoted greater birth weight. Those alleles didn't get passed down because those babies didn't survive to have their own children, right? So what happened over time was that over time, fewer and fewer babies were born that were really small or really large because those genes weren't getting passed down. We have stabilizing selection where the average of 7 is best, and so that is the size that is most commonly seen today. So reasons why it would not be beneficial to have a small or a large birth weight. If you've had children, you probably know even better than I do. But if you have very small birth weight, you're not going to be as strong, able to survive disease and different things, hardship in life. And if the baby is too big, you may have trouble actually being born, right? Getting out of the birth canal, and so the baby could die before it's even born. And so what we see is stabilizing selection for this average size. You're not too big, but you're not too small, just right. 7-pound baby. All right, so that is the last slide that I have for today. Again, the CHI-TESTer quiz is going to have material from chapter 21 and chapter 20. All right. And it's going to be just like the other CHI-TESTer quizzes. You're going to have a time limit, and you're going to have between today and midnight on Monday to take that test. Once you start it, you're going to have a restricted amount of time to take it. And again, I don't care if you look in your book, but you will not have time to look up and check every single answer. All right, best of luck. Please feel free to email me with any questions, and also best of luck on working on your lab reports. There is a quiz. It says something like lab report peer review reflection. It's not a quiz of your memory of anything. It's just a way for me to find out what are you going to work on in your paper. All right, so don't stress out about it too much. All right, I will talk to you later. Bye-bye.